It's an honor and a privilege to present this lecture in association with the Japan Prize Foundation's award, which I am pleased to share with Dr. Robert Weinberg. The title of my lecture will be Cancer Genes and Their Implications. And I want to first note that the work that I will present represents a group effort, a relatively large number of young people who have worked with me for many years, more than 160 of them, as postdoctoral fellows, research technicians, and doctoral students. I particularly want to acknowledge Dr. Kenneth W. Kinsler, who is standing in the front of this group, who has made many of the most important advances that our group has been able to accomplish. Now, I will divide this lecture into three pieces. First, the quantification of cancer genes, how many there are. Second, how we can exploit these cancer genes to help patients through the development of novel therapeutics. And third, how we can exploit this knowledge through improved diagnostics. And let's start with cancer gene quantification. There has been much confusion about the number of cancer genes, cancer driver genes there are, in part because of the very large number of studies that have been done in this field and the incredible amount of information that has been amassed over the past 10 years from sequencing entire cancer genomes, the coding regions of cancer genomes, and many other aspects of which have been revealed through these amazing technologies. And let's start with a single patient from pancreatic cancer patient PA34. Each of the chromosomes in this patient is represented by a kind of piano key, if you will, so they're 23. And each of the mutations in this patient that alter the coding region, that is, that change an amino acid from one type to another, are indicated by these black rings. And there are about 50 of them. But it's a bit simpler than that. If one just looks at the driver genes, one sees that there are really only 3. By driver genes, I mean those that are actually responsible for the abnormal growth that occurs in cancer through reducing cell death or increasing cell birth. One of these genes, the KRAS, is an oncogene. That's a gene that Robert Weinberg discovered long ago, and one of the things that he is being honored for in this ceremony. And the other 2 are tumor suppressor genes called TP53 and SMAD4. And the heights of these peaks represent their proportional representation in the population of pancreatic cancer patients. So nearly every pancreatic cancer has a KRAS gene mutation. About 3/4 of them have mutations in P53 gene, and about half have mutations in SMAD4, or genes that are related to these but in the same pathways. In contrast, the other genes that are mutated, these black tires, do not have any role in the pathogenesis of the disease. They're simply passengers, because every time a normal cell divides, 3 to 5 gene mutations occur in that cell. Those mutations are usually not in driver genes. They're usually in regions of the genome that cause no phenotypic defect. But if they occur in driver genes, in the right place within a driver gene, they can contribute to the process of tumorigenesis. And in 26 of the major cancer types, including all common solid tumors, there are only about 120 to 200 driver genes that have been discovered through a massive amount of sequencing. These are the major players. About 60% of these are tumor suppressor genes and about 40% oncogenes. And these genes represent less than 1% of the 20,000 genes that encode proteins and are present in the human genome. So it's really only a small fraction of genes which play major contributors to cancer. And this is really one of the fruits of the revolution in cancer research that has occurred over the last decades, the last 4 decades. And that revolution can be summed up as cancer is, in essence, a genetic disease. And it's not simply a single gene that's responsible for any cancer. It's a series of mutations. And in this diagram is summarized some of the mutations that occur in various cancer types. In general, it requires at least 3 mutations to convert a normal cell, say a normal epithelial cell in the colon or the pancreas, to a malignant epithelial cell. The first mutation, let's call it strike 1, by analogy to a baseball game, is the initiation of the process. When that process initiates, a small tumor forms, small and benign, too small to be seen by the naked eye. But over time, it can grow, still to a size that's often only microscopic. But then if one of the progeny of these cells acquires a second mutation, the cells can expand more into a visible lesion, a benign tumor that can often be seen by the naked eye. But still benign, it still does not have the capacity to metastasize, to invade. That invasive step is the key to cancer and what distinguishes cancer cells from benign tumor cells. The cells can invade through the basement membrane, invade through the vasculature, either the venous or lymphatic vessels, and migrate and seed other organs, causing metastases. Another important thing that has been learned through the molecular genetic analysis of cancers in our lab and labs throughout the world is that this is not a speedy process. It takes decades to get from that first initiating mutation to the last mutation. And these 3 sequential mutations, it's no game. This is what results in cancers that kill people, especially the metastatic cases. Almost everyone who dies from cancer dies from metastatic disease because the cancers were discovered too late to be cured by surgical excision or excision coupled with radiotherapy and chemotherapy. And that will be one of the themes that I emphasize in this lecture. Catching cancers earlier is what I believe is one of the keys to reducing deaths from cancer in the future. Now let's go on to therapeutics. And I'll tell you about a relatively recent story that's based on a genetic understanding of cancer. This landmark paper was published by Suzanne Topalianasked and her colleagues about 9 years ago, when they showed that an immunotherapeutic agent, an immune checkpoint inhibitor, an antibody against the PD-1 molecule, could result in remarkable responses, even in advanced cancer patients. And she noticed that patients with melanomas responded extremely well. Patients with lung cancers often responded, but patients with other tumor types generally did not respond. Now, Suzanne works in the Oncology Center at Johns Hopkins, the same center that I work in, and she and her husband, Drew Pardole, who was the first graduate student that I ever supervised way back in the 80s, came up to our lab and asked us if we could think of a reason based on tumor genetics that melanomas, patients with melanomas and lung cancers would respond, but patients with other tumors would not. And we looked at these tumor types, and we immediately recognized that there was something different about melanomas and lung cancers than most other cancer types. This was after the first genome-wide sequencing studies had been done, and this occurred in 2013. So on the y-axis here is noted the total number of coding mutations, that is non-silent mutations present in various cancers, shown on the x-axis. And just like in that one pancreatic cancer, PA34, that I showed at the beginning, the average number of such mutations in an individual cancer is about 50. Varies from cancer to cancer, of course. But look at these three cancer types. One is a melanoma, and two are lung cancers. And these three cancer types have many more mutations than other cancers. The reason, of course, is that melanomas develop in part through the mutations that develop through exposure to sunlight, ultraviolet light. And lung cancers, either non-small cell carcinoma or small cell lung cancers, develop generally through exposure to the mutagens and chemicals within cigarette smoke. So they have many more mutations than do the rest of the cancers. Now, we thought that perhaps this is the reason these cancer types, or patients with these cancer types, responded so well to immune checkpoint inhibitors. They had more mutations, therefore more chances to develop neoantigens, that is, proteins that have never been seen by that patient's normal cells, be recognized as foreign, and therefore an immunotherapeutic that can, in a sense, rev up the immune response could exert, at least in theory, a significant therapeutic response in these patients. Furthermore, we predicted, this was not yet demonstrated, that one other cancer type, colon cancers, called mismatch repair deficient cancers, that had a defect in their ability to repair mistakes, errors in DNA, would be even more sensitive to immune checkpoint inhibitors than melanomas or skin cancers. And those tumors are shown here. They have an average of 500 to 1,500 coding mutations per tumor, which is many times, 10 to 30 times more than present in colorectal tumors which have intact mismatch repair gene function. And we set out to do a clinical study to see whether this hypothesis was true. And the studies now have been published after several years. They were led by Luis Diaz and Young Lee at Johns Hopkins, and the results were really remarkable. As you can see from this plot, most of the patients with colorectal cancers who had mismatch repair deficiencies, tumors shrunk. And not only colorectal cancers, but many other cancer types had responses to immune checkpoint inhibitors as long as they had these large number of mutations due to a mismatch repair deficiency. As a result of these studies, the FDA approved immune checkpoint inhibitors, antibodies against PD-1, for the treatment of patients who had cancers that were mismatch repair deficient. And the FDA noticed that this approval marked a new paradigm in the development of cancer drugs. This was the first time that the FDA had approved drugs that were designed for any cancer, no matter what type, not just breast cancers or colon cancers or prostate cancers, but any cancer type, as long as it had a particular genetic characteristic, that is, lots of mutations due to mismatch repair deficiency. And this was a very wonderful example of precision medicine. Now, I want you to notice on this slide that although these patients underwent remarkable remissions, sometimes long-lasting, they were often usually not cured. And in fact, the inability to cure tumors is characteristic even of the new generations of therapies available to the clinician, whether they be targeted therapies or immunotherapies. So, for example, here is a patient with melanoma before treatment with a drug that attacks a mutant BRAF gene. You can see the dozens of tumors on this poor patient's skin. 4 months after treatment with this drug, all of the tumors have disappeared. This was truly miraculous, and this was one of the first examples of a targeted drug, a drug that targets the mutation. The drug has little effect on cells with normal BRAF genes, but if a tumor cell has a mutation in BRAF, it can have a remarkable response. Unfortunately, just 2 months later, the tumors recurred, all of them, at exactly the same place as they were before, as you can see from comparing panels A and C. Now, why is that? Why did these tumors recur, and why do they nearly always recur after some months, perhaps years of targeted therapies? The mathematicians that we have worked with, in particular the group of Martin Nowak at Harvard, have helped to answer that question. The fundamental basis for drug resistance is that there are already hundreds to thousands of resistant cells present in each clinically evident metastasis prior to therapy, even before any therapy begins. And those cells which are resistant prior to therapy will eventually result in the regrowth of tumors. The other cells within metastatic lesions will die, but these won't, and over the course of months to years will repopulate the tumors. Now, this brings into question the reason that we can cure mice. And all cancer researchers are very good at curing mice. I am very good at curing mice. If you are a mouse and have cancer, particularly one that I gave you a few weeks before, come to me. I can offer you hope. Unfortunately, I usually can't do the same with patients. But why is it that we're so good at curing mice and not so good at curing humans with advanced tumors? Some might say, well, the mice aren't really good models for human cancers. But I think that the genetically engineered mice that most current researchers use are really quite like human cancers in many respects. I think there's a simpler explanation. Here's a CT scan of a patient with metastatic disease from pancreatic cancer in a typical phase 3 clinical trial. All of those dark lesions represent individual metastases, and each of them contains tens of billions of cells. Now, I juxtapose a picture of a mouse in the CT scan, and you can see that individual metastatic lesions are larger than the whole mouse. If you just look at the mouse tumor and realize that unlike in this two-dimensional picture, tumors are 3 dimensions, you'll see that the tumors in humans are hundreds to thousands of times larger than typical tumors that occur in mice. So the reason, or part of the reason, that it's so much easier to cure mice is simply because there's so many fewer cells, and therefore fewer cells that are resistant to the drugs that we use prior to therapy. How to cure cancer? Turn people into mice. And I mean that only somewhat sarcastically, which I will tell you about in a few minutes. And this gets us into diagnostics. Now, historically, diagnosis, that means the earlier detection of cancer, has been thought of as separate from therapeutics. And there has been this ongoing debate about the best way to reduce cancer deaths. Is it through prevention or cure? I think that dichotomy is not really germane to the real situation. We need both, and they actually synergize, as I'll show you in a minute. Small amounts of disease can often be cured by conventional chemotherapy. These are the poisons that have been historically used to treat cancers for decades. And here is a graph showing patients who have micrometastases. That's small metastases. These are stage 3 colorectal cancer patients who are treated with conventional chemotherapy. And as you can see from this graph, nearly half can be cured. I don't mean just put into remission. I mean cured of disease forever. In contrast, if you look at patients who have visible metastases, on the right of this graph, none of them, or essentially none of them, can be cured by the same chemotherapeutic agents. And what that shows, again, is small tumors, metastases, which are present because eventually the disease recurs, can be cured, whereas larger metastases, those visible on CT scans or other imaging modalities, cannot be. And the same is true for the most advanced forms of immunotherapy. Immune checkpoint inhibitors, which I mentioned before, are a good example. Patients with relatively low tumor burdens can generally not be driven into long-term remissions by these immune checkpoint inhibitors, while patients with high tumor burdens can. I said that wrong. Patients with low tumor burdens can be driven into long-term remissions often, whereas patients with high tumor burdens cannot be. And another modern form of immunotherapy based on CAR T cells, it's the same. Those with high disease burdens cannot be driven into long-term remissions with CAR T cells, whereas a major fraction of those with low disease burden can be. And this gets us back to curing mice. Earlier detection of cancers when the tumors are small, when there's a relatively low tumor burden, can make human tumors more like those of mice. All therapies, whether they're immunotherapies, targeted therapies, conventional chemotherapies, will work better when patients are diagnosed at an earlier stage of disease. This makes the development of new drugs to be more effective in patients with lower amounts of disease. And it's why earlier diagnosis and the development of new therapeutics are synergistic. They go hand in hand. Now, we have been working on this idea of using the genetic alterations to detect cancer earlier for many years, actually close to 30 years. The first paper was published in Science in 1992, and eventually led many years later to the first non-invasive DNA screening test for colorectal cancer using DNA in the stool. More recently, we have worked very hard on developing blood tests for cancer. And the work I'll discuss now has been spearheaded by an incredibly talented MD PhD student named Joshua. In a paper published a few years ago, he showed that a test which he and his young colleagues developed could detect many early stage cancers. The test involved only 16 driver genes. Again, a manifestation of the fact that there are relatively few driver genes that have been discovered in human cancers, and even a small subset of them, 16 to be exact, can detect at least one mutation in many tumors. Now, that doesn't mean they can be detected in the blood of these patients, but if one looks at the tumors, one can see that this panel detects more than 80% of cancers. And that was coupled with carefully thresholded levels of 8 protein biomarkers that have been long used for cancer diagnosis. And in the original paper, Josh was able to show that a blood test called CancerSeq could detect more than 60 or 70% of cancers relatively early stage, that is non-metastatic, in many of the 8 cancer types developed. In fact, in 7 of them, and in 5 of those, ovarian, liver, stomach, pancreas, and esophagus, there was no other screening test available. Now, this was a retrospective study. We used only patients who already had been known to have cancer. What's needed, what was clearly needed then, was a prospective study. And that has been recently carried out by Josh and Marie Lennon and many colleagues who I'll show you later. But this was DETECT-A, a prospective interventional study that was carried out at Geisinger Health in collaboration with our group and with members of Thrive. And it involved 10,000 women, 65 to 75 years of age. There were no exclusions to entry other than a previous cancers. And those who had a positive blood test were then evaluated by a diagnostic PET-CT test to localize and confirm the existence of the putative cancers. And we were able to answer 5 questions through this study. First, can a multi-cancer blood test prospectively detect cancer in individuals whose cancer was not previously detected by other means? Seems rather simple, but extremely difficult to do, and had not really been done in an organized way in prior studies. And the answer to that question was yes. Each of these dots represents a cancer in a patient who is not known to have cancer, but whose cancer was first detected. The second question was, can such a test be incorporated into routine clinical care and added to the cancers detected by standard of care screening, particularly mammography and colonoscopy and other techniques for detecting colorectal cancers? And the answer to that is yes. Again, if you look at breast cancers, most breast cancers were detected by standard of care mammography. A few were not, and these could be detected by the blood test. Similarly, with colon cancers, some were detected by colonoscopy, others by the blood test. But in all the other cancer types for which there's no screening modality available, only the blood test was able to detect them, at least initially. A third question was, can such a test be used to intervene in the tumor progression process, leading to therapy with intent to cure? And the answer to that question is also yes. All of the cancers indicated in red here, which are a subset of the total cancers detected by the blood test, had therapy with intention to cure, usually surgery. Fourth, can this test or ones like it be performed safely without encouraging a large number of futile invasive follow-up tests, which is one of the major, if you will, side effects of any blood test. Too many false positives. And the answer to that was also encouraging, as you can see from this pie diagram. Of all the patients that were screened, the ones indicated in green, which are almost all of them, had no further procedures in follow-up. There was a very small number who had minimally invasive follow-up, and an even smaller number who had surgical procedures for non-malignant tumors. And in those 3 patients, they all had reasons for surgery, even though they did not have malignant cancers, malignant tumors. Those reasons are shown here. And 5, is this test clinically useful? conferring more benefit than risk to the screened population? And the answer to that is we don't know. DETECT-A was designed to assess safety and feasibility, not clinical validity. That will require a different kind of trial with far more than 10,000 individuals, which is being planned now. But this question can't yet be answered and can only be answered through a randomized clinical trial, not one that like this, which was designed to test safety and feasibility. Now, what I think is a reasonable goal for the future with respect to screening is listed on this slide. Right now, about 20% or less of cancers that occur in the US are detected by screening. The other 80% are detected by symptoms or signs of cancer, when it's often too late. I believe that in the near term, say within the next 5 years or so, it will be possible to make 75% of cancers detected by screening. Now, of those 75%, 2/3, I hope, can be detected by some sort of liquid biopsy, those that we or others have developed, and the other 25% by conventional methods such as mammography, colonoscopy, and the like. But together, if we can detect 75% of cancers by screening rather than symptoms, that will hopefully mark a turning point in the landscape of cancer and how patients are managed. So, in summary, much is now known about the genes that drive cancer. There are relatively few genes that in aggregate, when mutated sequentially, result in malignancy. There is, of course, much work to be learned, much to be done about how these genes work, and many in the cancer research community are focusing their efforts on learning about the functional consequences of these mutations, because that will be critical for designing new therapeutics that attack these mechanisms and the pathways through which these mutant genes work. Now, although cancer is far from optimally managed now, too many people, way too many people die from cancer, the history of medicine leads to optimism. Once a disease is understood, even in part, not necessarily in whole, it's generally only a matter of time before measures can be designed either through prevention or treatment that reduce deaths from that disease. And the same will undoubtedly be true for cancer. Not overnight, but certainly in the decades to come. And the final point that I want to make is that I strongly feel that two avenues of research must continue to be performed in order to really make the major impact on reducing suffering, morbidity, and mortality from cancer. Earlier detection combined with new therapeutics is what I believe is the key to reducing cancer deaths in the future. I want to acknowledge the companies that we have worked with to develop some of the tests that I've mentioned. And I want to thank the Japan Prize Foundation again for this singular honor to our group, and thank all of you who are watching this lecture for your attention.